in many cases we would like to highlight the interactions between a protein and a ligand. Here a ligand interaction diagram with a small molecule is shown. Here the ligand is shown in an R-atom representations, hydrogens omitted for simplicity, and most of the residues in the binding site are shown in this schematic simplified representation. Only one residue that makes critical hydrogen bonding contacts with the ligand is shown in a more detailed representation here. It's important that similar to 2D topology diagrams, such ligand interaction diagrams also do not capture all three-dimensional aspects and details of the binding interaction. They are designed to highlight the most important interactions and to show the residues that make up the binding site in order to understand why, for example, a specific mutation might influence or disrupt the binding interactions. You can see that even the ligand is three-dimensional entity, and the stretched out 2D representation is already a simplification. You can imagine that the protein binding site which surrounds this ligand is not a flat thing again. Right. We can have other interaction diagrams here. These are between two proteins, the SARS-CoV-2 spike protein and the AC2 receptor. You can see here that we have a mixed representation with cartoons for both proteins, and the most important residues participating in the interaction are highlighted with an all-atom representation, again without hydrogens, and you can see that the ionic interactions and hydrogen bonds are highlighted with these dotted lines. This is not the Omicron variant, this is the original one. The Omicron can make even more interactions with EC2. But here in this diagram you can have a glimpse of the actual three-dimensional organization of the binding site. You can see the shape of the surfaces that bind to each other. Let's switch to nucleic acids a bit. Nucleic acid structures can be also represented at different levels. There are 2D topology diagrams like this, and in the top you can see here the hydrogen bonds between the bases are shown in these lines. This is a double helix RNA helix, and this is a loop structure. This together is usually referred to as a stem loop structure in RNA biology. The linear representation depicting this is the so-called dot bracket notation. You can see that we have brackets here and dots, and it's important that the nucleotides that are positioned in the matching brackets are actually hydrogen bonded here. Okay, so you can see that this guanine and this uracil are hydrogen bonded here, and this uracil and this guanine are also hydrogen bonded here. So the matching brackets indicate hydrogen bonds between the two nucleotides. These topology diagrams for nucleic acids are very commonly used, especially for complex RNA structures. Again, they do not, because they cannot show all three-dimensional contacts, and there can be parts that are not to scale. For example, in this diagram you can see these very long connecting lines. These are obviously just for depicting purposes in order to be able to place these structures here to show the most important contacts. This is something just for the illustration that shows you that these are actually linked to each other covalently. Such diagrams usually emphasize the hydrogen bond pattern, and very limited information about the 3D contacts is involved. So this does not mean that we can't have a three-dimensional contact or even several hydrogen bonds between, for example, this loop and this loop. Okay, so it might be that in the three-dimensional organization we have additional contexts. These topology diagrams can give us a good overview on the we call it secondary structure of RNA, the hydrogen bonding pattern. These are the 3D representations of nucleic acid structures. Like for proteins, the all-atom representations are of limited use. We usually use a representation in which we show the sugar phosphate backbone or the backbone with a line or something like that. Here you can see that the sugar, the 5-carbon sugar, is shown with a pentagon, and the bases are shown in a simplified geometric representation. So the purine and the pyrimidine scaffold is shown, but not the atoms. So you can have a basic view on how the base pairs are oriented relative to the backbone. On the right, it is a even more simplified representation where the bases are only shown as rods. Okay, so just or sticks. So such a stick corresponds to a full nucleotide, if you like. This is again just to show how the base pairs are oriented within the structure. Okay, so these are highly simplified representations to show you the major characteristics of the structure. Here the coloring is by chain, and here the coloring is by structural unit of the RNA. This is basically DNA structure, which goes behind the classical to stranded helix. This is a DNA enzyme or deoxyribozyme which has catalytic activity. The last topic is to represent dynamic features of proteins. Proteins are not rigid entities. They have different movements on different time scales, and sometimes we would like to depict this in order to understand the functional role of these internal motions. We can have linear representations, of course, when we depict a dynamic parameter along the sequence, just like here. This is here in this diagram we compare 4 different domains, domains that are truncated at the C-terminus, and domains that are free or in complex with the peptide. These are PDZ domains, and the parameter is a so-called order parameter, which ranges from 0 to 1, and 0 means more intensive motions or more flexibility. 1 means rigid structure. You can see that in these several regions have increased motion, and this is different between some of the structures. This is very good for a good overview and for a comparative purpose, but this doesn't give us any hint about the nature of the motions, or does not show us the location of the interesting sites or different sites in the 3D organization. In order to have a better depiction of dynamics, we usually have to do a bit more serious data analysis. Without going into much detail, there are methods which we can generate structural ensembles. That means that not one structure but multitude of structure, several structures. The diversity of each structures reflects to some extent the experimentally determined dynamical parameters of the proteins, or we can run a molecular dynamic simulation and have a representation of the estimated dynamics of the protein. And if this is not enough for us, we can analyze these ensembles with methods like principal component analysis, which will highlight us the most important motions that contribute most to the observed variability of the structures. So here the previous 4 domains are shown the motions of the previous 4 domains are shown in a principal component analysis plot. You can see that several variants occupy different regions of this PCA plot. So this gives us more information about the extent of the motions, whether the parameters observed in one protein can be numerically similar to the other protein, but the interdependence of these parameters and the full structure might mean that the actual region in the conformational space that is represented by the molecule is different. Okay, so here we can identify the type or nature of the given motions and to what extent the given proteins occupy the region along the given motion. So here the conformational space is basically simplified by this principal component analysis here into 2 dimensions, and using the results of this analysis we can generate structures that represent extremities along the given motion. It should be noted that here you can see that here this loop is moving. That means that this principal component 1 means that this loop is moving along with some other motions here, but the extent of these motions in these particular depictions can be scaled in order to show the nature of the motions more clearly. So here it does not necessarily mean that actually the motion is like this. It shows us the nature of the motions, how what is the main characteristic of the given motion. It is also true for the second principal component here. How does such a structural ensemble look like? This is another example of ubiquitin. We have a number of structures here. Here some over 100 structures are depicted here. The coloring scheme is according to the S-square order parameter, and it shows that we have a relatively rigid core of ubiquitin, and the C-terminal end of the protein is quite flexible. And again, we have a principal component analysis here. Here along the first 4 components, 1, 2, 3, 4, and the red dots correspond to this ensemble-based representation, and the black triangles correspond to X-ray structures of ubiquitin. And the purpose of this plot is to show that the conformational space explored by the solution state dynamics of ubiquitin covers the conformational space represented by the known X-ray structures of ubiquitin. And this has biological consequences in terms of binding. So it means that all the conformations that are observed in ubiquitin with bound partners are basically adopted by the free protein, and the partners can select what they bind best to. Last but not least, we can also represent the motions with the so-called porcupine plot or arrow plots, where we basically put a cone or an arrow on the C-alpha atoms, and that represent both the direction of the motion and the extent, the amplitude of the motion. These are again prepared usually based on principal component analysis. So usually such a plot describes a motion along one of the principal components in the protein. The take-home message of this lecture is that first you should specify the purpose of your visualization. Will it just be a nice colored image on the cover of a magazine or a scientific journal? It's very nice if it is, but then this is the main purpose of the visualization. Or is it crucial to understand the biological importance or a function of a system? Okay. In this respect, is the overview of the structure, the full organization of the structure important, or you would like to highlight a specific detail, and you have to be clear what the main message of the visualization is. I usually tell my PhD students when we write a paper that each scientific paper usually contains one or two key figures, key images that depict the main message of the paper. And if this is a structural related message, then you have to be well aware of what you would like to communicate to the readers. And then when you know this, you should choose a concept, what to do, and there are several aspects. For example, do you need a structure-based analysis to show what you want to see, or you can depict directly the structure itself, or you need to do some calculations to show it. Can the given feature well represented in a 3D representation? Is it needed, the 3D representation, or is it better to simplify, to use a 2D topology plot? Okay, maybe you should it's enough to use a linear representation along the sequence, because that shows your purpose best. Okay, and what is the coloring scheme you would like to use? How would you like to emphasize the most important aspects? If you use different types of plots in the same figure, it's strongly recommended that you use the same coloring scheme, like I have shown you for SARS-CoV-2 proteins, for the spike protein, when the linear representation had the same coloring as the structural representation. Do you need your visualization in a comparative assessment? Is it better shown along the sequence, or you should highlight the structure? These are very important aspects to consider, and I hope that this lecture gives you at least some initial hints on how to start this. Thank you for your attention.